Hello, my name is Edwin Oh, and I am an associate professor at the UNLV School of Medicine and the Nevada Institute of Personalized Medicine. Thank you all so much for joining this presentation today. I would like to share some of our short stories about SARS-CoV-2, the discovery of variants in Southern Nevada and also in Arizona in a talk entitled Identification of Highly Transmissible SARS-CoV-2 Variants in Sewage Predicts Clinical Disease Burden in the United States. So before I begin, I'd like to certainly acknowledge all of the different collaborators who made this work possible. First and foremost, I'd like to acknowledge Dr. Dan Garrity and Dr. Katarina Papp at the Southern Nevada Water Authority. If it weren't for both Dan and Katarina, none of this work would have been possible. Both of them really helped us understand the potential of wastewater and the surveillance of pathogens like SARS-CoV-2 using wastewater. Together with Dr. Walter Betancourt over in the University of Arizona in the Water and Energy Sustainable Technology Center, we were able to use many of our tools to characterize wastewater genomes coming directly from a building such as a university dormitory. And with folks at the Southern Nevada Health District and also in the public health labs in University of Nevada, Reno, we were able to juxtapose the wastewater genomes with clinical genomes so as to appreciate how well we could use wastewater to complement the surveillance of COVID-19 in Southern Nevada. And I'd also like to acknowledge funding that made this work possible through NIH, the folks in Nevada and the governor's office, and CDC. So I get to sit here and talk about all of the wonderful findings that folks in the lab really developed and thought about and spent many, many different days and nights on better understanding. And most of this work here was driven by a talented postdoc, Dr. Van Vo, here in the middle in the green, and she is now currently a research assistant professor in the lab. The work was also very much supported by undergraduates in the lab, by graduate students, by clinicians, by bioinformaticians, and by other data scientists. And together we were going out to manholes at elementary schools and sampling wastewater so as to bring these different resources back into the lab and process them for viral material that we analyze for absence and presence of the virus, and also more specifically for different variants. I'm also director of a genome analysis and acquisition core, and here Shirley Shen is the lab manager, and Dr. Richard Tillett is the bioinformatician who really built the pipelines that were required to analyze all of the data that I will be talking about today. So in the first short story, I'd like to discuss how we use whole genome sequencing analysis of wastewater samples and how this analysis was sufficient to predict the types of human infections that we were getting for COVID-19 in Nevada. So here on the left of your screen, in this figure, what we're depicting here in each panel is really the different facilities that we were examining across Southern Nevada. I've only listed 7 of these different facilities, and most of these facilities here really services up to 2.2 to 2.3 million people down here in Las Vegas, Henderson, and really Southern Nevada. And if you look specifically at panel B in this slide, what we are plotting here on the left y-axis is the number of new daily cases in this sewage shed per 100,000. And what we're plotting here is this line in red, where we see a decrease and an increase and then a decrease. And over here on the right side of this graph, I'm plotting SARS-CoV-2 concentration, wastewater SARS-CoV-2 concentration in gene copies per liter. And this is really a depiction of the different points here in the blue. And all of this data, the clinical cases and the wastewater data, is plotted from the end of July of last year to May of this year. And the point of this panel and really of this figure is that when clinical cases increase, we also see wastewater viral content increasing. And when we start to see the decreasing in cases, we also see the decrease in viral concentrations in the wastewater. And all of this data was done by Katarina Papp and Dan Garrity over in the Southern Nevada Water Authority. So using these different data points, we wanted to then better understand whether these increases were driven by specific variants. Back in November and December of last year, many of the countries that were suffering from COVID-19 experienced a surge in a variant known as the Alpha variant, or B.1.1.7, or the variant first discovered in the United Kingdom. We wanted to better understand whether some of these variants were really driving these surges. So we use this workflow here on the right for next generation sequencing. And what we came to understand was that at least 10 nanograms of complementary cDNA was required before we could then make these libraries. Now, this was true for wastewater samples and also for clinical samples. The type of library preparation kits that we use were a combination of either a hybrid capture library prep kit that you can purchase from Agilent and or Illumina. And we also used a combination of different amplicon-based kits from Paragon Genomics, from Swift, and from QIAGEN. And our analysis of all of these different library prep kits was that certain kits had better coverage of different parts of the genome. And that's part of the reason why we ended up using these different kits. We also wanted independent verification of our samples by using two different library prep kits on the same sample. If we were to see the same variants show up using these two different library prep kits, we would then have more confidence of the results. We then sequence our samples on an Illumina NextSeq 500 instrument, and we either use a mid or high output flow cell, depending on whether we had more samples or less samples. So for a mid output, we could load this flow cell with about 100 samples, 100 wastewater samples or clinical samples, and expect about 1 million reads per sample. Whereas a high output flow cell, we could load up to about 400 samples and expect about, again, 1 million reads per sample. And the type of metrics that we were hoping to achieve was greater than a 50x depth or greater than an 80% coverage. So what 50x depth means here is that every nucleotide had at least 50 reads on average. And out of the 30,000 nucleotides, we could cover more than 80% of that entire genome. And we were looking at a minimum threshold of about 5% allele variant frequency. So these metrics are a lot more stringent than what one might use for a clinical genome. Typically, a clinical genome or a genome from a human infected by COVID-19, we wouldn't really need such metrics because there's only one genome present in a human, most of the time infected by SARS-CoV-2. In wastewater from a facility, if you imagine a 10% positivity rate in an area that's made up of about 100,000 people, we would now be expecting anywhere between 10 to 100 to 1,000 different genomes, viral genomes in that wastewater sample. And so we would want to ensure that there was enough reads to cover all of those different genomes. So if you can predict that there are 100 genomes present in your wastewater, you really want to have a minimum depth of about 100x so as to cover on average all of those different genomes. So using these metrics, we then analyzed all of the different facilities and we sequenced the wastewater genomes. So in this figure, what I'm depicting here is a cartoon of the SARS-CoV-2 genome, where all of these different boxes correspond to the gene cassettes that are present in the genome. So many of you are probably familiar with the spike protein cassette, and what's listed here are all of the different mutations that have been associated with the B117 variant. So these variants here, these mutations here also are characteristic mutations that are typically found in B117. So when we now look at positive controls, we would expect to see all of these different mutations present. So for example, in RNA from a patient who has been infected by B117, we can demonstrate that all of these different variants are present. We can also take a synthetic genome, an RNA that's made in the lab, and we can also identify all of these different mutations. And this gives us specificity and sensitivity type of calculations for our sequencing technologies. Now, an interesting finding here when we analyze all of the different wastewater facilities is that we identified B117 as early as December 21, 2020 in one of our facilities. We could identify many of the mutations that are typically found in B117. And as time went by, we could see many of the different facilities lighting up with B117 across time. However, a key point here is that we could identify this genome, this B117 genome, as early as December 2020. Given that we can do this for B117, given that we can identify these genomes, this genome, B117, we naturally wanted to know whether we could identify other genomes. During this time of December, January, February, another variant was quite predominant in Southern Nevada, also known as B.1.429, or the variant that was first discovered in California. When you look at the cartoon of the genome, here are mutations that are typically found and associated with this variant that was first found in California. And interestingly enough, again, we were able to identify this genome as early as December 14, 2020, and again in December 28 of 2020 in different facilities. And across time, we start to see more and more of these types of genomes showing up in Southern Nevada. So two major take-home points here from this slide is that you can use wastewater samples to identify specific variants. And equally important, we were able to identify B117 or the alpha variant as early as December, and also the B1.429 genome as early as December of 2020. Now, when you juxtapose the wastewater genomes with the clinical genomes, you can better understand how these wastewater genomes may or may not be predicting the emergence of these different variants. So when you look at samples across December 2020, most of these genomes were either a 20A or a 20G genome. We did not identify a human infection in December 2020 that corresponded to B117 nor to B1.429. These genomes started to show up in early January 2021, where here in this brown colored part of the pie, we start to see B1.429.427, and we also start to see B117 here in this blue. 2% of all cases were B117 in January. The same was true for February. We start to see an increase in March and also in April. The same is true for B1.429, where we start to see a lot of it in January, more of it in February, March, and April. An interesting feature is we were not identifying these genomes back in December. And one important reason that that may be the case is that many of the labs were not sequencing genomes back then. So we think that the wastewater genomes can actually give you so much more precision if you do not have the resources to be testing people on a regular basis. Now, given that we could do this, we could run this wastewater variant analysis for alpha and B1.429. We naturally have done this for delta, lambda, mu, and we've been able to identify all of these variants. So a key takeaway point from this slide is that wastewater surveillance can indeed predict the emergence of these variants, depending on what type of public health resources are present in a certain geographic area. Now, a similar theme here is that because we can also identify SARS-CoV-2, we can identify many other pathogens in these wastewater samples through next generation sequencing. And that's exactly what we've done by looking at antimicrobial resistant alleles in communities in Las Vegas. And we can start to see the emergence of a number of these different alleles. And we saw a surge of these alleles back in December of 2020. So we're putting this paper together now. But the bottom line here is that you can identify pathogens from wastewater with this new tool of next generation sequencing. Now, given that we can do this analysis for large treatment facilities, we naturally then wanted to ask whether we can do this for manholes. And so in a second short story, together with Dr. Walter Bencourt over at the University of Arizona, we worked together to sequence wastewater samples coming from surveillance of 20 different buildings last year across the University of Arizona campus. Walter and the group at the West Center wanted to know whether building surveillance could be an early warning system for infections in dorms, and it worked really, really well for them. They were able to identify a signal from the wastewater, and then subsequent testing of individuals within the dormitory led to the identification of 3 infected students. We then took that wastewater sample and then we sequenced the corresponding wastewater sequence, and we found at least 2 different signatures within that wastewater sample. To us, that's a really important discovery for a number of different reasons that I've summarized here in this cartoon. Remember, with Walter's study, they were able to use wastewater qPCR results, a positive hit here in the wastewater, to then go into the dormitory to identify infected individuals. What we would like to propose is that you can not only identify the absence or presence of a pathogen, but you can now sequence the genome of that pathogen. And the reason you'd want to do that is that you can now go back to that infected individual and sequence that corresponding sample too, so as to match up these genomes. And the obvious reason why we want to be able to do this is we want to identify the index individual. We want to know who's the source of this genome so that we can quarantine the individual and be able to mitigate any risk of infections. So the reality is that in the future, we are truly hopeful that we can really go we can proceed in a manner where not only do you go from wastewater to the infected individual, but now take that genome and compare with that wastewater genome. Along a similar thread, we've also, in the third short story, used wastewater sequencing to better understand the impact of COVID-19 across a certain community. So take Southern Nevada, for example, where here in yellow in this zip code, we find very few cases versus here in this different zip code, as an example for, again, Southern Nevada, where you find very high case counts. If you look at income levels, you'll find that there are correspondingly lower, on average, medium household income, whereas here you find higher income levels. We wanted to better understand whether in these differences and demographic differences and viral differences, were we seeing higher viral counts in elementary schools? Now, elementary schools, the analysis of elementary schools is, for us, was really a function of there not being vaccines for children less than 12 years. So we wanted to better understand, right? Were we seeing higher numbers of infections in one area of Las Vegas versus another? And the interesting observation was that it didn't matter whether we were analyzing schools in the north or schools in the south of Southern Nevada. We were still seeing viral concentrations. Now, certain schools had more viral concentrations than other, but the bottom line is that back in June of 2021, we were seeing viral concentrations regardless of median income levels. We think that this is potentially going to change over the next year. And so we're analyzing this data really for the next few months to better understand whether there are going to be new health disparities that emerge. One thing for sure is that either children or adults at these elementary schools are infected and therefore shedding virus into this wastewater. So in conclusion, most of these findings have really led us to start thinking about a new project, an urban pandemic preparedness project, where we're examining wastewater facilities, we're examining dormitories, schools, nursing homes, and also we're examining airports, hotels, stadiums, so as to better understand what types of pathogens are growing, are emerging here, and what type of pathogens are being imported, either from domestic locations across the country or from international locations. And we want to be able to develop the most appropriate processing workflow so as to get to the bottom of this answer. We're also developing new probes and new next generation sequencing technologies to analyze these samples and the appropriate pipelines, the bioinformatic pipelines to share with the scientific community and also, very, very importantly, to share with the public. With that, I'd like to end this presentation and I would be happy to take any questions.